Hello and good afternoon friends, welcome once again to the CEC EDICET live lecture. Dear friends, in this session today we are going to discuss about cells and organs of the immune system. We are going to discuss how the cells and organs function as well as how they develop. We would also be going to touch on a very important issue nowadays, that is on the human stem cell research. For this, we have again with us in our studios Dr. Amit Bhattacharya. He is an assistant professor in Ramjas College, Delhi University. So first of all, I would like to welcome our guest, Dr. Amit Bhattacharya. Hello sir, welcome to the EDICET lecture.
Thank you, ma'am.
When we talk about the immune system, lots of questions appears in our mind, but now we are today discussing how cells and organs basically function, and I hope that through this lecture we would be able to understand how basically they contribute for the immunity or the immune system.
Yes,
so over to you sir.
Thank you, ma'am. Thank you very much. So good afternoon dear friends. So today I am going to present the presentation on the cells and the organs of immune system. Now I will be basically touching upon what exactly and how are the different cells produced in our body which functions in the immune response mechanisms, and what are the organs in which these cells develop and matures, and subsequently I will be going into how there is a coordination between the cells and the organs of the immune system. So the immune system plays a very important role in the body mechanisms. Like suppose we suffer from some pathogenic attack from the virus, bacteria, fungi or parasite. These are the immune system cells which continuously circulate within our body through the blood vessels or through the lymphatic vessels, and they immediately recognizes these antigens or the pathogens in our circulation, and subsequently they clear them off from our body immune system. So if these diseases are not cleared from the body conditions, then the person will be suffering from lots of syndromes, which happens in many of the chronic diseases which occur for a long period of time. So we will be talking about various cells, how these cells are formed, what are the types of these cells, and subsequently what are the organs in which these cells are developing. So before starting into, let's define what exactly a immunity is. So the immunity is a state of protection from the infectious diseases. So the immunity, if we say that this person has got immunity to this particular pathogen, it shows that the person has got the immune system cells circulating within its body for that particular pathogen. Like suppose we see the picture over here where the bacteria have infiltrated the body circulation system. So we can easily see over here that there are some immune response cells. What these immune response cells are doing is they are immediately engulfing this bacteria and they are recognizing this bacteria so that they don't spread out throughout the body mechanism. So the immunity is one of the basic mechanism through which these pathogens or these antigens are cleaved up and they are diminished from our body circulation. Now, what are the basic cells of the immune system? So we know that there are various blood cells which circulate within the blood vessels, like the red blood cells, platelets, which are very important in the blood clotting part. Then comes is the leukocytes. So these leukocytes are broadly divided into number of subclasses which has got lymphocytes, there are different forms of lymphocytes are present. Then comes the monocytes, which subsequently goes on to form the macrophages, and lastly comes are the granulocytes, under which the neutrophils, eosinophils and basophils come. So these leukocytes are in total called as the white blood cells, and they are the one which are responsible for the immune system mechanisms which develops or grows within our body circulation. Now, what are the various organs of these immune system responses? So the organs have been broadly divided into two major categories. The first one is called as the primary lymphoid organ, and the second one are called as the secondary lymphoid organ. So the primary lymphoid organs are the organs which are present within our body, and these are the organs which provide the appropriate environment for the development and maturation of these white blood cells or the lymphocytes. And the secondary lymphoid organs are the organs which play the second role. They trap the antigens which are circulating within our blood circulation, and subsequently these are the sites where the maturations of the lymphocytes also takes place. So these are the sites where there is an effective interaction of the white blood cells and the antigens or the pathogens, and subsequently these pathogens are killed and they are evaded out from our body circulation. Now both of these organs are connected to each other or coordinated to each other with the help of the blood vessels and the lymphatic system. So we'll go into each of these cells as well as the organs and the lymphatic system, how exactly the coordination thing is going within our body circulation. Now the first process which starts where most of these red blood cells are generated is called as the hematopoiesis. As the name is suggesting, hemato means the blood or the heme, and poiesis is the generation. So hematopoiesis is the mechanism by which there is a formation and development of red and white blood cells. So the red blood cells, we know that are made up of the erythrocytes, or which are the oxygen carrying molecules within our blood. The other part which are present in the blood circulations are the white blood cells, under which comes are the lymphocytes, granulocytes, and the monocytes or the macrophages. And finally comes is the platelets, which are very important, plays a very important role in the clotting mechanism. So all these cells are produced from some of these cells which are present in this bone marrow. So we can see that most of the bones, the long bones like the femur bone, or which is also known as the thigh bone, contains a medullary cavity, and in this medullary cavity there is a marrow which is present. So this bone marrow is responsible for the generation of all these red blood cells, white blood cells, and the platelet. So these bone marrow contains certain cells which are called as the hematopoietic stem cells. These cells have got the property of self-renewal, as well as they can differentiate to form any of these cells like the red blood cells or the white blood cells or the platelet. So it all depends upon what are the growth factors and what are the other protein molecules which are present at that particular time, and that will guide these bone marrow hematopoietic stem cells to go into that particular lineage and form that particular cell. So the bone marrow plays a very important role in the development of the immune response. So many a times we see that the patients in which the bone marrow have suffered, the patient becomes immunocompromised, means the patient doesn't have the immune system or a well-versed immune system to fight with the external pathogens which are invading in his body. So these bone marrow are very critical and very important for the development of these immune system. Now let's take a closer picture of how exactly a human bones looks like. So if we take a bone anatomy, we can see that the bone anatomy which has been sliced up, we see that there is a medullary cavity, and inside that medullary cavity there is a bone marrow which is present. In this bone marrow there are certain cells which are present, which are called as hematopoietic stem cells. As the process is called as hematopoiesis, that's why the stem cells are also called as the hematopoietic stem cell. Along with these stem cells, there are stromal stem cells which provides the appropriate microenvironment and the nutrition for the growth and the development of these hematopoietic stem cells. Now, what exactly are these hematopoietic stem cells? So hematopoietic stem cells are the multipotent or pluripotent cell which has got the ability to self-renewal ability, as well as it has got the ability to differentiate into various forms of the cells, like it may develop into a erythrocyte, granulocyte, monocyte, lymphocyte, or platelets which are formed from the megakaryocyte. So they can form a variety of cells, and they are very important for the immune response things. The second major important point of these hematopoietic stem cells is they are in very few numbers. Only one hematopoietic stem cell per 50,000 cells in the bone marrow is present. So you can just imagine the low amount of hematopoietic stem cells. So if you see the image over here, we can see a round kind of cell which is the hematopoietic stem cell, and which is surrounded by the maturedly formed erythrocytes or the red blood cells. So these hematopoietic stem cells are very few in number. Now, what exactly is the process of hematopoiesis through which there is a formation and development of the red blood cells or the white blood cells. So the hematopoietic stem cell, as I told you, has got the property of self-renewal, so it can go on to form two of the lineages or the progenitor cells, which is called as the myeloid progenitor cell and the lymphoid progenitor cell. This myeloid progenitor cell can further go on to form the dendritic cell, then it can go on to form the monocytes, which subsequently goes to form macrophages. It can also form the granulocytic cells, which contains the neutrophils, eosinophils and basophils, and it also forms the platelets, which are formed from the megakaryocytes. So these are very essential for the development, and the myeloid progenitor cells are essential for the development of erythrocyte, because they go on to form the erythrocyte progenitor cell, and subsequently these go on to form the RBCs or the red blood cells. Now the other part of these hematopoietic stem cells can form the lymphoid progenitor cell, which can go on to form the lymphoid cells, which are broadly classified into B cells or the B lymphoid cells and the T lymphoid cells. So we will come into each of these cells one after another, and we will see that how important these cells are for the immune system or the immune development mechanism. Now how exactly these hematopoietic stem cell goes about from the fetus to the adult form. So during the developmental process or in the early weeks of development, the hematopoiesis takes place in the embryonic yolk sac. So in the embryonic yolk sac, the hematopoiesis development start taking place. Subsequently it is taken over by hemogenic endothelium, and when the fetus develops into a few more months and few more months time, so these hematopoiesis stem cell process is then taken over by the fetal liver, which plays a major role in the hematopoiesis in the fetus. And when the fetus grows into the adult, this hematopoiesis process is taken up by the bone marrow, which are present in the bones. So this is how the embryonic to the adult hematopoiesis takes place, and how the progression takes place with the development of the hematopoiesis and the immune system within the organism. Now the hematopoietic stem cell research have gained lots of momentum over the period of time, and now the hematopoietic stem cell research have made it possible to study this hematopoiesis in the in vitro conditions or in the laboratory conditions. So if we have a petri dish which contains the appropriate growth mediums and the development nutrients, and there is a layer of stromal cells, and if we put one of the bone marrow into these petri dishes, these bone marrow stem cells automatically go on to develop the progenitor or the perfectly formed cells, depending upon the growth factors which are present in that particular medium. So the growth factors and the cytokines, which is again a protein molecules, are essential to stimulate the formation of hematopoietic stem cell colonies in the bone marrow culture. So like this way, like these studies have found out two of the major growth factors and the cytokines. First is called as the colony stimulating factors, which is called as the CSF. As the name is suggesting, it has got the ability to induce this cell division and cell proliferation within the hematopoietic stem cell. And the second major factor which have been found through this hematopoietic stem cell research in the laboratory condition are the erythropoietin or the EPO. These growth factors are developed or produced by the kidney cells, and what they do is they induces the terminal development of the erythrocytes or the red blood cells and regulates the production of the red blood cells. So these erythropoietin growth factors are very essential for the development and normal functioning or the production of the red blood cells. Now these stem cell research have got lots of clinical uses, like if we have got a human pluripotent hematopoietic stem cell, we can easily form a bone marrow, or if we have got a pluripotent stem cell which can go on to form the nerve cell, heart muscles, pancreatic islets. So we can easily treat the patient who is suffering from some kind of disease. So we can replace these disease or damaged tissues with these newly formed things. Like suppose a person is suffering from the insulin diabetes or the insulin, so we can easily replace the deficient or the inactively working pancreatic islet cells with the newly formed stem cells, pancreatic islet cells, and the patient can recover from that thing. Like the patient who suffers from the cardiac arrest or cardiac problem, if there is some problem in the heart muscle cells, so we can easily generate these heart muscle cells from the in vitro human pluripotent stem cell and replace them into the patient where there is a damage or the disease conditions has occurred. So there is a absolutely high value of these stem cells which is gaining a lot of research attention throughout the world, and the world is now focusing upon developing these stem cell research to replace the disease or the damaged condition cells. Now, the hematopoietic stem cell research have been also instrumental in the cancer therapy. Like lots of time we hear that the patient who is suffering from the cancer goes about for the radiation or the anti-cancer drugs. But these radiation and these anti-cancer drugs have got lots of side effects, of which one of the side effects is the targeting the hematopoietic stem cells within the bone marrow. So the bone marrow stem cells gets distorted or gets damaged by these radiations and the anti-cancer drugs. So the recipient who is getting those things becomes immunocompromised, or the immune system in these particular recipients decrease down. So it is very essential to replace this hematopoietic stem cell and transplant a freshly induced and healthy stem cells into that particular recipient. So the healthy stem cells are taken up from a healthy donor from the bone marrow, and then they are injected into the recipient bones so that they can replace the damaged bone marrow stem cell, and the patient can recover from the after effects of the radiation or the anti-cancer drugs, and the patient will develop the immune system mechanisms, and subsequently the patient won't be immunocompromised, or he or she will be able to fight with the infectious disease which is causing a bad health to the patient. Now, these hematopoietic stem cells are essentially controlled and taken up at a particular point with the help of a mechanism which is called as the programmed cell death. So we just know that the red blood cells or the erythrocytes have got a lifespan of about 120 days, after which these red blood cells are killed or gets damaged automatically. Now, how exactly these things are happening? So all these cells are having a particular lifespan, after which it starts getting into the aging process, and once it ages up to a particular limit, it is replaced from that particular cell or that particular organs with the production of new or the fresh cell which replaces those things. So it has been estimated that about 3.7 into 10 to power 11 white blood cells are produced or generated each day. So you can just imagine how there is a replacement process which is going within our body, in which the damaged or the old cells which are going old because of the aging are being replaced by a fresh inflow of the cells, and this is how the programmed cell death process takes place. Now, what exactly a programmed cell death process is? The programmed cell death process is broadly called as the apoptosis because there are distinct morphological changes which occurs in the cell surface or within the cell. Like suppose a cell over here which is shown on the left hand side panel, this cell has to go into the programmed cell death or the apoptosis process. Now what happens is there are some kind of morphological or structural changes which start occurring in these cells. Like the example is the chromatin or the DNA start getting clumping up or getting compact. The second major thing which occurs is the cytoskeleton or the surface of the cell becomes distorted, and what happens is all these phenomena make the cell non-viable, so it is not living a proper way which it has to. So what happens is subsequently it goes on to cause the nuclear fragmentation, or the nucleus get fragmented, and subsequently there are apoptotic bodies which are formed, and these bodies what they do is when they are released from those particular cells, they are immediately phagocytized by the macrophages or the immune system cells. So they are engulfed by these macrophages and they are taken up within these macrophages, where various lytic enzymes, what they do is they digest up these particular apoptotic body, and they release these aging cells out of the body. So this is how this cell goes about for the programmed cell death. So this programmed cell death plays a very important role in maintaining a homeostatic or a steady state conditions within the hematopoietic stem cells also. So it is very essential that the programmed cell death functions properly. Now if you see the picture over here, I have shown a picture how the apoptosis occurs in the thymocytes. Thymocytes are the T cells which are developing in the thymus, which is again a primary lymphoid organ which we will be discussing right now. So if you see the picture, there are normal thymocytes. So the electron micrograph is showing the normal thymocytes, where we can say a fully formed or a matured thymocytes which is there. As soon as the cell moves into the apoptosis, we can see that the cell become distorted. There are some structural changes which are occurring. The nuclear fragmentation is also occurring, and subsequently what happens is these apoptotic cells are then taken up by the macrophages which are circulating within the blood circulation, or when they go into the lymph vessels, they are easily taken up by the lymphocytes and they are degraded and released from these mechanisms. So the apoptosis plays a very important role. Now what exactly happens to the cell which doesn't go for a normal programmed cell death because of aging, but has got some kind of injury which is caused to it? Like suppose some kind of injury occurs to you in which some of the cells have got disrupted, or some of the cells which are not functioning properly, how these cells goes into a cell death. So this process in which the cell death of the injured cells occurs is called as the necrosis. The necrosis is very different from the apoptosis, because in this necrosis process the injured cells swells up. So it increases its size, and subsequently what happens that most of the organs or the organelles which are present within these cells swells up, and finally it ruptures out or bursts out to release the contents to the exterior environment. As the material is released to the environment, what happens is there is an inflammation which is occurring, and an inflammatory response occurs because of the release of the intracellular components. Now in the case of apoptosis, there is no inflammation because all these fragmented bodies are engulfed into apoptotic body, which are further engulfed by a macrophages, so that there is no inflammation process which occurs in the body. So what we see is many a times when we get some kind of injury at some of the places like cut or something, we see that there is a immediately redness or some kind of inflammation which is occurring at those particular part because of the cell death of the injured cell. So the necrosis process has started up, so all the immune system cells have started coming to that particular position, and the inflammation and the degradation of these cells start occurring, or the pathogens start occurring. So the necrosis also plays a very important role in the homeostasis of these cells. Now let's come into the first type cells of these immune system, which are broadly called as the lymphocytes. So the lymphocytes constitute to about 20 to 40% of the body's white blood cells which are circulating within our body. And it has been said that in the humans there are about 10 to the power 11 lymphocytes which are present within the human body. And these lymphocytes plays a very important role in the immune system because they have got two of the most characteristic features. The first one is the diversity, specificity, and the other one is the memory. So the memory part is very essential for the lymphocytes. Now these lymphocytes have been broadly divided into three major populations. The first one is called as the B cells, the second one is called the T cells, and the third one are called as the natural killer cells, and they are being divided into these category based upon their function and the cell membrane components. So we will come into these ones one by one. Now how exactly these lymphocytes gain the momentum when the antigens or the pathogen invades the body? So what happens is, in a normal conditions, these lymphocytes are present in the G0 phase, which is the resting or the quiescent phase. As soon as an antigen enters the body circulation, so what happens is these B cells can immediately recognize these antigens, they get activated, and as they get activated, they get into the cell cycle. So the cell cycle arrested is now being removed. So the B lymphocytes now goes into the cell cycle where the G1 phase starts, where most of the genes get activated. Subsequently, the S phase starts where the DNA synthesis is occurring. So the lymphoblast S lymphocytes are formed. So if you can see the electron microscope diagram, we can see that there is a huge amount of nuclear DNA which is being replicated and formed at this particular phase, and finally these replicated cells divide into two daughter cells, each containing an exact copy of the parental cells. So now these daughter cells have got two fate. Either they can go on to form the effector cells which immediately attack the antigens, and the second fate is the memory cells where they remain within the body circulation for a long period of time in the quiescent phase, and as soon as there is an invasion of this particular antigen again into the body circulation, they immediately recognizes that there is a memory cells which are present within the body circulation, and what happens that these memory cells get activated and they clear the second antigen attack within the body. This is what exactly is happening in the vaccine strategy. So the vaccines are given so that these memory cells and the effector cells are produced. The effector cells clears out the infectious disease from the body, and the memory cells remains within our body circulation, and as soon as these cells get a fresh follow-up of these infections within our body, these memory cells get activated and clear them off from the body. Now the B lymphocytes, the first form of the lymphocytes are called as the B lymphocytes. The name has come up from the position or the organ where the maturation takes place, or the development and the maturation takes place. In the case of the birds, it takes place in the bursa of Fabricius, while in the humans and the mice, the maturation of the B lymphocytes takes place in the bone marrow. So we can see the organs in the picture, where in the birds it is taking in the bursa, while in the humans it is starting up in the bone marrow cells. Now the B lymphocytes have got a distinguished feature of a membrane bound immunoglobulins which are present on the surface of these B cells, and they act as the receptor for the antigen, and it has been estimated that about 1.5 lakh molecules of these antibody receptors are present on a single B cells. So in a single B cells there are about 1.5 lakh molecules of antibodies which are present. Now the B lymphocytes, how exactly they function? So what happens is, as soon as a antigen invades our body, which may be a foreign protein, virus, bacteria, parasites or a fungi, so what happens is these B lymphocytes which are circulating within the blood circulation or the lymphatic vessels, they immediately recognizes these antigens, and as soon as they recognizes these antigens, they go into the division process where the effector and the plasma cells are formed, and these plasma cells start secreting the antibodies which go and recognizes these antigens, and subsequently they felicitate the clearance of these antigens or the pathogens from our body. So this is how the B lymphocytes function in clearing up the pathogens from our body. The next lymphocyte cells are called as the T lymphocytes. As the name is suggesting, the T word has come from the site of maturation of these T lymphocytes, which is the thymus. This thymus organ is present on the top of the heart. So like the B lymphocytes, these T lymphocytes also have got membrane receptors for the antigens, and most of these T lymphocytes recognizes the antigen which when and are presented on a specific protein which are called as a major histocompatibility complex, then only they can aggregate and they can develop into a mature T lymphocytes and immediately attack the thing. So over here I have shown the structure of a T helper cell, which is a subpopulation of the T lymphocyte, and a T cytotoxic cell. So in both of these you can see that there are TCR, which is the T cell receptors which are present, and in the T helper cells there is a CD4 receptor which is present, while in the TC cells, which are T cytotoxic cell, the CD8 receptors are present. Now how exactly these receptors and these cells functions when a antigens invade our body? So like an antigen which invade our body. So what happens is these antigens are then engulfed by the specialized cells, and what they do is they process and fragment these antigens, and they present these antigens on a small molecule which is called as the MHC molecule. So if you see the left hand side picture, there is a presentation of the antigenic part which is the epitope on a class 2 MHC. So as soon as these specialized cell which has engulfed these pathogens, they process and present a small part of these pathogens on the surface of it with the help of the MHC, and with the MHC these T helper cells immediately recognizes and they become activated. As the activation process starts, there are production of some cytokines which occurs. So all these small ball like molecules are the cytokines which acts as a major factor in the activation of the other cells. Now what exactly happens in the case of the T cytotoxic cell? So what happens is the antigen from these pathogens are processed and presented in the case of TC cells in the form of MHC molecule, MHC class 1 molecule. So in the MHC class 1 molecule, these antigens are processed and presented, and these TC cells immediately recognizes this class 1 MHC molecule presented cells, and what they do is they activate these T cytotoxic cells, which are further activated by the cytokines which are released by the T helper cells, and what they do is they coordinately work to clear this antigens from the blood circulation or the body part. So these T cytotoxic cell plays a very critical and very important role in the clearance of these pathogens or the infectious diseases from our body. Now in this lymphoid cells, there are other cells which are present which are called as the natural killer cells or the NK cells. So if you see the image over here, the natural killer cells is showing killing and destroying a tumor cell. So the cell on the left hand side is the natural killer cells which can be seen interacting with the tumor cells, and these natural killer cells expresses a membrane bound receptor which is called as a CD16 molecule receptor, which plays a very important role in targeting these tumor cells or the virus infected cells, and they kill or clear these cells by a process which is called as antibody dependent cell mediated cytotoxicity. Now there are various conditions, syndrome conditions which have been found in the humans which are related to the deficiency or non-functioning of the immune system mechanism. One of the condition is called as the Chediak-Higashi Syndrome, which is a very rare and autosomal recessive disorder which is found in the humans. So if we see the image over here, the neutrophils or the immune system cells are showing the abnormal conditions. So in this particular syndrome condition or the patient, what happens is the patient suffers from defective immune killer cells functioning, and there is a deficiency of neutrophil activity also. So the patient develops the chances of development of leukemia and lymphomas. So the cancer formation increases in these particular patients because of the absence of an appropriate and effective immune system mechanism. Now the second class of cells of the immune systems are broadly classified as the mononuclear phagocytes. All these cells have got the property of phagocytosis. So there are two subclasses under this particular category which are called as the monocytes, which are circulating in the blood circulation, and the macrophages which are present in the tissue forms. So what happens is granulocyte monocyte progenitor cells which are formed during the hematopoiesis, when they leave the bone marrow, they are formed as the promonocytes. Subsequently, as they enter the bloodstream, what happens is they mature to form the monocytes. Now these monocytes circulate within the blood circulation, and subsequently they migrate into this tissue or the tissue spaces where they develop into specific tissue macrophages. Like there are certain macrophages or the immune system cells which are present in the lungs, livers and kidney. So these macrophages plays a very important role in saving these particular organs from the pathogenic or some disease conditions attack. Now the first mononuclear phagocytes under this one is the monocytes. So if we can see the structural thing over here, we can see that there is a nucleus, there are lysosomes which are formed. These lysosomes are the organelles which are responsible for the clearance and the cleavage, because these contains the lytic enzymes which are responsible for degrading the phagosome or the pathogen which has been engulfed by these monocytes. Now the other monocytes which develop in the tissue organs are called as the macrophages. Now these macrophages have got the pseudopodia. These are the special things which are present in these cells, through which these cells engulf the antigens or the pathogens which are there in the blood circulation or the lymphatic vessels or the tissue condition. So these are engulfed through these pseudopodia, and subsequently the phagosomes are formed, to which the lysosomes, which are shown over here in blue in color, this fuses with these phagosomes to release its lytic enzymes which also contain bactericidal enzymes. So what these enzyme does is they have got an acidic kind of environment, so they degrade these pathogens or the antigens, and subsequently they are released off from the circulation. Now if we see some of the macrophages which are present in different tissues and are serving different functions. So the first macrophage over here is the alveolar macrophages which are present in the lungs. Then there are different macrophages which are present in the kidney, which are called as the mesenchymal cells, and in the liver these macrophages are called as the Kupffer cells, and in the connective tissues they are called as the histiocytes. So these macrophages plays a very important role in clearing and detecting these antigens from these critical organs like the lung, liver, kidney and the connective tissue, and subsequently after recognition clearing them off from the condition so that these organ doesn't suffer from any disease condition or some organ failures. Now how exactly the phagocytosis process takes place? Like the phagocytosis process is the process in which the macrophages ingest and destroy the pathogen. So over here we can see a macrophage which is ingesting or phagocytizing the bacteria which are present in the near proximity of its environment. So the phagocytosis is the process where these macrophages ingest and destroy these pathogens. Like what exactly happens in this phagocytosis process? If we see the picture over here, the bacteria are engulfed with the help of the pseudopodia. Then these engulfed bacteria are digested upon in forming a phagosome. Subsequently to these phagosomes, the lysosomes fuses. These lysosomes, as I told you, contain the lytic enzymes. So what happens is these lytic enzymes capture the material and subsequently degrade and digest these things, and the digested product is then released from these macrophages. So this is how the phagocytosis or any of the bacteria or the pathogens which are circulating and which has been recognized by these macrophages are engulfed and cleared from the circulation. Now the next cells of the immune system are called as the granulocytes, which are broadly divided into neutrophils, eosinophils and basophils depending upon the stain which they are taking. Like the neutrophils can be stained with the neutral stains, while the eosinophils get stained by the eosin stain and the basophil gets stained by the basic dyes or the stains. So these neutrophils constitute about 50 to 70% of the circulating white blood cells, and they are the most essential part of the circulation because neutrophils are the cells which are the first cells to arrive at a site of inflammation or at the site where the injury or any kind of problem which has disease condition has occurred within our body. So the neutrophils are very critical and essential. Then comes the eosinophils, which are about 1 to 3%, or the basophils, which is only 1%. Now the neutrophils are also called as a polymorphonuclear leukocytes because of their multi-lobed nucleus. So we can see the structure over here and we can see a stained image of a cell in which we can see a multi-lobed nucleus which is present. Along with these multi-lobed nucleus, there are granules which are present, which are primary and secondary granules, which contains the lytic enzymes. So these neutrophils have got the phagocytic property. So what they do is they phagocytize these pathogens or the antigens like what we discussed, and they are taken up by the phagosomes, and subsequently the granules release up their enzymes into these phagosomes and they are destroyed from the circulation. The next comes are the eosinophils, which are shown over here, contains the granule, and they also contains a lobule or lobule nucleus. The third granulocytic cells are the basophilic cells, which are shown over here. They contain lots of granules which are present, but these basophil doesn't have the phagocytic property, so these are present in a very low quantity within the blood circulation. Another important cells, immune system cells, which are important for the development of allergies and inflammations, are the mast cells. So these mast cells can be found in the skin, connective tissues of various organs, mucosal epithelium tissues of the various organs like the respiratory tract, urinogenital tract, and the digestive tract. Now these mast cells plays a very important role in the development of allergy and the asthma. So allergy and asthma, which we regularly hear these terms. So what happens in this asthma is, as soon as an allergen, over here we have taken the allergen as the ragweed, what happens is when they enter the body circulation, they are immediately recognized by the B cells which are circulating within the blood circulation, and these activated B cells then mature and develop into the plasma cells, where there is a production of ragweed antibodies or the IgG antibodies are produced from these plasma cells. These plasma cells then release plasma cells antibodies, then go and attaches to the mast cells receptors which are present on the top of it, and these ragweed then bind to these antibodies and elicit a response which is called as the IgE primed mast cell response, where there is a release of pharmacologically important molecules from these mast cells, which causes sneezing, running nose, watery eyes, and lots of disease conditions which are normally associated with the asthmatic attack. Now, the organs of immune system has been broadly divided into two major categories. The first one are called as the primary lymphoid organ, and the second one are called as the secondary lymphoid organ. So the primary lymphoid organs are the organs where the development and the maturation of these lymphocytes takes place, while the secondary lymphoid organs are the sites where there is a trapping of these antigens or the pathogens takes place, and it provides the site for the mature lymphocytes to interact with these pathogens. Now let's see what are the organs of these immune systems. So if we can see over in this diagram, the bone marrow and the thymus, which is present on the top of the heart, is the primary lymphoid organs, while the secondary lymphoid organ contains the Peyer's patches, which is present in the intestine, spleen, lymph nodes, and the tonsils, acts as a secondary lymphoid organs. Now the primary lymphoid organs are, the first primary lymphoid organs are the bone marrow, where the hematopoiesis process takes place, in which the red blood cells, white blood cells, and the platelets develop. So the second primary lymphoid organs are called as the thymus, which are the sites for the development of the T cells and the maturation. So this is a flat bilobed organ which is situated above the heart. So if you see the diagrammatic representation over here, there are dividing thymocytes or the maturing thymocytes which are present, which subsequently goes on to maturation process and then are released into the lymphatic vessel for the body circulation. Now we see that with the age, the immune system or the person's immune system decreases, because the thymus size also decreases with the age. So as soon as the thymus start developing from the birth till the 10 or the 20 years of age, it gains the maximum maturity and subsequently it decreases down. So thymus are the organs where the T cells maturations takes place, and T cells are very critical in evading and clearing these pathogens or the immunotoxic molecules. Now, the secondary lymphoid organs contains the lymph nodes which are present throughout our body. So, these lymph nodes are the specialized organs which helps in trapping the antigens from the local tissue. So, if we see the picture over here, the lymph nodes are attached to the lymphatic capillaries which are present within the tissue spaces, and from these tissue spaces it is capturing the antigens or the pathogen. So, from these tissue spaces, these antigens or the pathogens are taken up into the lymph nodes which contains the lymphocytes, and these are the sites where there is a active interaction between the antigens and the antibodies. And this is a structural representation of the lymph node, how exactly the lymph nodes looks like, which contains the cortex, paracortex and medulla, and how the development process of the lymph node takes place. The secondary lymphoid organ which is important is called as the spleen. So, the spleen specializes in filtering the blood and trapping the blood-borne antigens. So, the spleen plays a very important role in filtering the blood. So, all the bloods which are circulating within our body goes into the spleen where the filtration process takes place, and from this filtration process all the antigens or the pathogens are taken up by the spleen, and in this spleen there is a clearance or the degradation of these pathogens or the antigens takes place. The secondary lymphoid organ, the MALT, plays a very important role, which are called as the Mucosal-Associated lymphoid organs. Under this mucosal lymphoid organs comes the mucous membrane which are lining most of the systems like the digestive, respiratory and the urinogenital systems, and the MALT lymphoid organs contains the Peyer's passages and the tonsils which were prominently here in this case. So, the tonsils plays a very important role as the secondary lymphoid organs because there are three forms of tonsils which are present. So, if you see the picture over here, there is a palatine tonsils, there is a lingual tonsil which is present on the top of our tongue, and the third pair of the tonsil is called as the pharyngeal tonsils, which is present in the pharyngeal place. So, these tonsils also plays a very important role in the lymphoid processing and the immune system mechanism. Now, the human immune system is broadly based on two major components. One is called as the innate, the other one is called as the adaptive immune response. So, the innate is the less specific component of the immune system, while the adaptive component is the more specific immune system which compromises all these immune system cells which we just discussed. So, these adaptive immune response have got a high degree of specificity and remarkable property of memory. So, wherever there is again an inflow of these particular pathogens into our body, there are memory cells which are circulating within our body. They immediately recognize these things and they filter out from the body circulation. So, with these things I end the presentation over here, and subsequently we will discuss later on what are these, how these functionings are taken place and how these immune system mechanisms are being taken up in the research conditions, and where the person suffers from immunocompromised property and how these immune systems are replaced in those particular patients or the persons. Thank you very much.
With this note, thank you, sir. Thank you so very much for giving a deep insight into this topic. As we talked about the human stem cell research, nowadays we have seen that number of disease or ample of diseases could be treated very well with the help of the human stem cell. So, what is this process basically?
Actually, the human stem cells, studying the human stem cells or developing a organ or a cell from a stem cell is a very tedious task. It doesn't take only few days. There are lots of years and years of research, because the stem cell, the scientists have been able to take out these stem cells and they can develop or grow these stem cells in the in vitro condition or the laboratory conditions. But what are the growth factors which are responsible within our body? Why are these growth factors present, and what are the various other proteins like the cytokines which are present at that particular point, and how these stem cells are developing into these kind of specific cells is a very critical and very important question to be asked. So, the stem cell scientists are now finding out what are the growth factors and how these cytokines are helping the stem cells to develop in the particular cells. Like we just discussed about how the human pluripotent stem cells which have been taken from the umbilical cord or some of the part of the stem cells from the human patient, healthy donors. They have been cultivated in the petri dishes, and how the researchers are trying to find the growth factors through which these stem cells under these microenvironment, they develop into the nerve cells, like many of the patient who suffers from the abnormal nerve conditions or Alzheimer's or Parkinson's diseases, where there is a non-functioning of the nerve cells or the nerve conditions over there, how these damaged cells or the non-functioning cells can be replaced with a fresh flow of these flow cells which have been produced in the laboratory conditions, like in the case of the heart diseases, where the patient suffers from number of chronic heart diseases where the heart muscles doesn't function properly, how those damaged or non-functioning heart muscle cells can be replaced with the help of these stem cells developed cells or the heart cells. And this research is still going on. The stem cell research as of now can also be said that it's in a very early phase of its development. Still lots of development has to be done to know exactly how and why these things are happening within our body or a normal human being, and what are the factors which are responsible for the development. Only then we will be able to replicate these processes within the artificial condition or the in vitro condition, and once these cells are formed in the artificial conditions, we can easily inject or inflow the things. But the hematopoietic stem cell transplantation process has already been found very successful in replacement of the damaged stem cell from the bone marrow, and it has been found in number of cancer patients where these stem cell transplantation has taken place. The patient develops the immune system and doesn't remain immunocompromised, because most of the cancer patient who undergoes the radiation or chemotherapy becomes immunocompromised, and there is no immune system cells which are present in the body of that particular patient. So reviving those hematopoietic stem cells is very essential in those patients.
Definitely, sir. The way you have described all this methodology, I hope that all the viewers and the students who might be watching us would be aware of this particular topic. Now the question arises, though we have discussed a lot about cancers in our previous lectures, here we have talked about cells. Basically, if we talk about cells, and in the case of cancer, we used to say growth of abnormal cells.
Yeah.
What is the difference between a cell or an abnormal cell?
Okay. So during the presentation, I told you about a process which is called as the programmed cell death. As the name is suggesting, programmed cell death. So all these cells have got a particular lifespan after which it has to die by a process which is called as a apoptosis. Like for instance, the cells or the red blood cells or the RBCs which carry the oxygen throughout our blood and takes the carbon dioxide to the lungs, where there is all the processes which are going. So the red blood cells in our body has got a lifespan of about 120 days. So after 120 days, a cell, that red blood cell, has to die by the process of programmed cell death, which has been programmed for that particular cell. But many a times what happens in the patients like leukemia, where there is abnormal conditions which are developing, these red blood cells doesn't die because of the programmed cell death or the aging process. So they start developing and get accumulated in the body. So there is an increased level of these red blood cells or the immune system cells which develops into our body. So the programmed cell death is very critical in keeping a homeostatic condition. Homeostatic condition means the number of cells which are present within our body and the number of the cells which are getting killed or are going into the death condition. So the death condition cells which are going into the death or getting into the apoptotic process has to be replaced by equal quantity of these cells, so that there is a homeostasis which is present within our body. So if this homeostasis is imbalanced in any of the disease condition, the patient suffers from the abnormal growth. Like that's what exactly happens in the cancer condition. The cell doesn't go for the normal programmed cell death. So if the cell doesn't go for a normal programmed cell death, it will automatically go for a synthesis and the division process. So one cell which has to go to the death after 40 days. If it doesn't go to death after 40 days, or the programmed cell death or the apoptosis, after 40 days it will automatically die to form another 2 progeny of the cells. So that 2 progeny or the 2 daughter cells are extra cells within our body. So that's how the cancer develops, and now these 2 extra cells will further develop into 2 more cells. So like this way there is an exponential increase in the number of cells or the abnormal growth within the cancer patient. And many of the disease conditions where there are immune related cancer things, these cells, immune system cells, also develops in a high conditions or a high number, copy numbers in those particular cases.
This was the case with the disease such as of cancer. Now the question arises, is there any term, or do our body produce some more dead cells also?
No, our body doesn't produce any dead cells, but our body has got these immune system cells which takes care of these cells which are about to die. Like suppose a skin cell has to die about after, for example, after 6 days or 7 days. So as the aging process takes place, these skin cells automatically develop some kind of mechanisms or some kind of receptors or some kind of growth factor changes around its surface, so which immediately sends this signal to these particular immune system cells. So now what happens is, these immune system cells like the macrophages or the monocytes, which are continuously circulating within our blood circulation or the tissue conditions, they immediately recognizes these dying or the aging cells with the help of either the receptors or some kind of thing. So they give a so-called eat me signal. So that's called as a eat me signals, means now I am about to die, so come and eat me. So these cells, what they do is they immediately come to that particular position and they engulf and phagocytize this about to die cells or the dying cells by the process of apoptosis. So now these cells which are going into the apoptosis are being engulfed by the macrophages, and in the macrophages further the lysosome activated processes occur. The phagosomes now fuses with the lytic enzymes, and this causes the degradation of these cells, and when these cells get degraded, the byproducts or the digestive products are released out from the cells. So the apoptosis plays a very important role, which is known for the programmed cell death. But many times what happens is we have got some kind of injury, like suppose we get a cut in our skin, so that's an injury which has occurred to the skin cells or on the surface. So what happens that the second level of mechanism starts, which is called as the necrosis process, which we discuss. So what happens that in the necrosis process, the injured cells swells up and it bursts out from that particular part. Immediately the bursting causes release of the components, and there is accumulation of these immune system cells around that region, and many a times what we see that when there is a cut or something, there is a redness which is occurring at that particular point, and subsequently the redness, when the redness has gone, there is a pus cells which are formed. So these are the pus cells which are the white blood cells which have fought with the antigens or the dying cells, and these are the pus cells releases are the things which are the digested or the byproducts from these interaction between the antigens and the immune system cells. So these are very critical processes, and the understanding of these things are also very essential for getting into the immunological aspects, how exactly the body functions, what are the organs, and how these organs are protected from the exterior infectious diseases.
Thank you, sir. Thank you so very much for delivering such a nice lecture, and I would like to tell you all that you can access this particular lecture with the help of the YouTube also. We daily upload our lectures, and with the help of those lectures, I hope that you would be able to study more and more. Thank you, sir. Thank you so very much. And in near future also, we would be carrying more lectures of this type so that our students could be benefited. Thank you.
Thank you.